Please welcome the panel on Placing New Bets in Biomedical Innovation. Where is the next cure? Moderated by Senior Health Correspondent at Time, Alice Park.
It did sink, didn't it?
Okay. It would have been good if I just wiped out a bit. Yeah. We're going to Biotech News.
Good afternoon, everyone, and welcome. I am Alice Park, Senior Health Correspondent for Time, and it is my honor to be moderating this session. I think we can all agree that this is a really exciting time in biomedicine. Science, I think, we would generally say that science proceeds at a pretty slow pace, but in recent years, it does feel like it's been everything everywhere all at once. You know, starting with completely new technology for vaccines with mRNA. We have our very first FDA-approved CRISPR therapy. And of course, we have AI continuing to play a big role across the whole spectrum of how drugs are developed. So I am really thrilled because we have on our panel today many of the people who are really instrumental in those innovations. So I think we'll just start with having each of you introduce yourselves very quickly. Noubar.
Sure. I'm Noubar Afeyan. I'm founder and CEO of Flagship Pioneering, which is a company that starts biotech companies, and I'm also co-founder and chairman of Moderna.
Hi, I'm Aloe Blacc, singer and songwriter, recent biotech entrepreneur, founded a company called Major Inc., and we are working at the crux of ending all disease with small molecules.
Hi, I'm Daphne Koller. I'm an OG AI person at this point, having worked in the field for over 30 years. I'm the founder and CEO of a company called In Citro, which does AI for drug discovery and development.
Hi, everyone. My name is Sam Kulkarni. I'm the CEO of CRISPR Therapeutics. And at CRISPR Therapeutics, we take this platform called CRISPR to edit genes and tackle diseases. We have an approved drug for sickle cell and thalassemia, and we're working on a number of other diseases like heart diseases, diabetes, and cancers.
Hi, everyone. My name is Pablo Legorreta. I'm the founder and CEO of Royalty Pharma, a business that funds the R&D ecosystem, but also involved with 4 or 5 other companies where I'm chairman or shareholder, companies that are involved in cell therapy and also working with potentially Sam and CRISPR, editing potentially organs, which is really exciting.
And we've had some very exciting innovations in that, right, Pablo, very recently. We'll get into that. But I wanted to just start a little bit and put everything in context of these new innovations that we've been seeing, mRNA, CRISPR, some of the gene editing things that we've been able to do. For decades now, I think our therapies have been really based on medicinal chemistry, small molecules. People have to take these either in a pill or an injection, often daily, and if you're lucky, maybe once a month or something. But as powerful as they are, they're not perfect. So I think it seems fair to say, at least from my opinion, that we are at the dawn of a new era where we're looking at cell-based, gene therapy-based, and really more systems-based approaches to treating disease where we can have a more sustainable effect. So please correct me if I'm wrong, but would love to hear if that seems right to all of you.
Well, I mean, indeed, the first wave of small molecular protein drugs that made biotherapeutics is giving way gradually, not fast enough, to much more kind of both precise and, let's say, powerful approaches to both therapy and vaccines. And that's because we're beginning to understand the information nature of biology and nature in general, and therefore we're being able to go at it with information molecules. So things like mRNA, siRNA, some of the editing techniques, that's all entering the realm of information processing, in one case biological information, and in the other case computing that's helping us do that. So I do think this new wave of new modalities, however daunting they seem to an industry that hasn't seen a new modality previously for a long time, is becoming more the norm than not. It'll not replace small molecules and proteins, but certainly there's 3, 4, 5 now proven up-and-coming platforms. And so we're going to be able to aspire to do more and I think have more impact.
Yeah. And one of those is CRISPR. So Sam, let's talk a little bit about what it means to now have an FDA-approved CRISPR therapy. What sets CRISPR apart?
Yeah, happy to talk about CRISPR all day, but in a nutshell, CRISPR is this system that was elucidated from bacteria, where bacteria have an intelligent system to cut viruses, recognizing the sequence of the virus and defend themselves. And that was adapted to say, how can we change our own genomes? Just to contextualize all this, about 120 years ago, we discovered that there are things called genes in our bodies. We didn't know what caused heredity or what caused our features to go to our kids and everything else. And it took a long time from there to about the start of the millennium where we discovered what the entire sequence of our genome is, and we're still trying to figure out what we learned from it. And CRISPR allows you to fundamentally alter diseases or disease course by going to the root gene and making a modification there. Now, when we first started embarking on this journey, everyone thought it would take 20 to 25 years for a platform to become a reality. But what's so powerful about CRISPR is within 10 years of elucidating the platform, we now have an approved drug for sickle cell disease and thalassemia. And sickle cell disease, it's a terrible disease. People with sickle cell live with pain, chronic pain, but also acute crisis, go to the hospital all the time. We've known about this disease for 70 years, but no intervention has been developed or no medicine has been developed. And finally, now we have the opportunity for a cure because of this powerful platform that's CRISPR.
So to get there, though, and it's been a very relatively short time, scientifically speaking, as you said, in a decade to get from basic science, first paper published on a brand new approach, to having a therapy that's approved for patients in about 10 years is lightning fast, I think. Maybe not to other industries, but in biomedicine, I think that is pretty quick. But it didn't happen overnight because that work, there was a foundation of work that was laid in basic science and even with mRNA that happened over decades. So I want to talk now a little bit about how we bring that process up to speed and how we streamline that process a bit to get there, to get an example like a CRISPR in about 10 years instead of 50 or 60 years. So Aloe, you come to biomedicine from a very different world. You're a musician and artist. What brought you to biomedicine?
What brought me to biomedicine was identifying a technology at the University of Houston developed by Dr. Gomika Udagama-Soria at the School of Pharmacology. He's working in a class of molecules called peptoids, and I was looking for a host-directed prophylaxis approach really to SARS-CoV-2. We'd been reading a bunch of papers and thinking about how can we block the virus rather than just boost our immune system. Of course, mRNA vaccines were doing really well, reducing mortality. But I thought there probably could be another solution. And I found the university professors working in this, and I sponsored some research, some in vivo research that ended up being successful. Learned more about the platform and realized, wow, they can approach infectious disease, oncology, autoimmune disorder, and basically have a platform that can address multiple indications. And I want to try to help them translate that as much as possible. And really what it comes down to is, you were mentioning how can we more rapidly find solutions with some of these interventions. And I think there's a standard of research that is sort of the status quo, which is one bug, one drug. You kind of trying to identify a target, then you identify the drug lead, and you try to optimize that drug lead. There are new ways to do that. I think machine learning and AI is going to help us with that, but the bench is always going to be the bottleneck. You have to do the research in physical space. And Gomika Udagama-Soria has developed this amazing high throughput screening assay that identifies targets and drug leads at the same time, with the most highly target-specific drug leads. And I think that when it comes to any particular target that you're looking for, whether it's a receptor or a cell surface biomarker, he's able to identify a drug lead for it. And I think that is really important. Getting that part of the puzzle done earlier, faster, with less money and less time is really important.
And that's sort of where our model may be losing. Do you think a lot of opportunities? I mean, you saw one, but not everyone in your world or even in other worlds will see that and see the need there. So how do you, when you look at this world and the way it's structured in terms of how ideas get from like a lab bench, you know, to the next stages, what do you think is critically needed at this point?
What I think is critically needed is that the research dollars really have to be focused on finding the most highly target-specific drug leads or interventions, so that by the time you get into phase 2, phase 3 trials, we're not losing billions, sometimes hundreds of millions of dollars of work just because you didn't have the most target-specific lead in the first place. And I think that's a lot of time, a lot of money. Taxpayer dollars a lot of times go to that because we're paying our taxes to the federal government, which then gives money to NIH, and NIH gives the money to the university professors. It could be spent much more efficiently.
Okay, we're going to come back to that idea. Pablo, I want to talk to you because you also bridge the entire spectrum from basic science all the way to, you know, pharmaceutical companies who are developing compounds. Tell us a little bit about Royalty Pharma and your model for having an impact in this space.
Sure. So it started 27 years ago with an observation that so much money had been invested by National Institutes of Health in academic research, and realizing that universities were going to end up owning royalties on products that were discovered there. Also hospitals, medical research hospitals. And we started just monetizing these royalties, offering these institutions an upfront payment for their future stream of payments, royalties, that allowed them to diversify, allowed them to build buildings, reinvest in research more quickly. It then evolved in about 2012, 10, 12 years ago, where we said to ourselves, we have learned how to assess a commercial risk of a product because we would buy those royalties once the product was approved. So we're trying to just figure out how big it's going to be. But we started to make some investments in products that were approved, but then had additional indications, like Humira, you know, approved for RA, but then had the potential to be approved in many autoimmune diseases. And we started to then assess approval risk. So in 2012, we decided, let's just fund the biotech ecosystem and big pharma. And we've done that now at scale. So for example, last year, we looked at 400 potential opportunities, which is obviously much more than once a day. And then from those 400 opportunities, we did 9 transactions and deployed $4 billion into the ecosystem, funding many potential products. Over 10 years, we've invested about $25 billion, of which about 60% is in approved products and about 40% in late-stage products, phase 2 and phase 3, which is a decent amount of money, $10 billion invested into the ecosystem, funding biotech and pharma at a time where innovation is incredible and financing innovation has become difficult. So, it's great for these companies and obviously a great opportunity for us.
Tell us a little bit about that divide between the sort of good ideas out there and the resources to bring them in. Aloe mentioned this as well. So how many, or what is the kind of proportion of ideas that are lying fallow right now in like academic labs, findings that could become new treatments, but perhaps because there are lack of resources either on the scientist's part, in terms of knowing how to take it to the next step, or funding that they can't raise?
So what you're talking about is not so much once a product gets into human clinical trials, but really translating the academic science into products. And obviously, it's a very difficult process, but it's a process that over time, over the last 10, 20, 30 years, has gotten a little bit more efficient. And I think actually the person that can really speak to that is probably Noubar, who his businesses is actually coming in at that stage of this whole development process. We come a bit later.
Okay. So Noubar, let's talk about mRNA, because it's easy to look back now that we have these successful vaccines, but at the time when Flagship sought out mRNA, there were no products, nothing approved, no guarantee that this would even turn out to be a viable platform. What was it about that technology that stood out, the features of that stood out to you as something that could have an impact on new therapies, the next generation of new therapies?
So what you're talking about in terms of taking academic research and making it into a commercial reality usually starts with either a problem that you're trying to solve or a solution you're trying to find problems for. In our case, we don't approach it that way. We try to imagine what could exist and try to figure out how to make it real. And in this case, the simple question we were asking ourselves back in 2010 is, what if you could introduce a molecule into a patient and make any protein you wanted? That was the what if question. We didn't start with RNA, we didn't start with a disease. We just started saying, what if you could do that? So what? Well, the so what would be that the entire biotech establishment of billion-dollar facilities and 10 years of product development and process development would essentially be consumed by just the body doing it. You'd still have to do clinical trials, but it's a very different kind of value proposition. Then if you say, okay, how do I do that? So this is how our process worked, going backwards from a desired possibility back to a reality. And if you look at all the molecules you could use, you could put DNA, you could put RNA, you could put proteins that somehow figure out how to reform themselves, you could put phage. So there's a lot of different approaches. Of those, mRNA seemed like a molecule that on the one hand had the capabilities, like the software of a cell, but on the other hand, people had basically tried in academic labs and given up. Why? Because despite the modifications that had been made before 2010, for which a pair of scientists just won the Nobel Prize, when you went into animals, it just wasn't enough of a reduction in the innate immune response. So as entrepreneurs, as scientists, we had to say, okay, how long will it take and how much money will it take to actually go from that point to completely reducing the innate immune response? And for that matter, how do we get it into the right cells? No answer to that back in 2010. We set out because we thought the prize would be big enough and that it would be ubiquitous. If you could do it once, you could do it for a lot of different applications. And so a lot of the work, what is usually viewed as kind of an afterthought, if you think about it as a scientific project, was actually done in this company, Moderna. 10 years later, when COVID made Moderna famous, people thought, well, this is completely untested technology, which, while true from a commercial standpoint, we had spent $2.5 billion building a platform that at the time COVID showed up had 20 clinical programs, 10 vaccines, 10 drugs already being tested, many of them through phase 1. So yeah, it was an overnight success with $2.5 billion and 10 years in the making. And you have to have the willingness to basically follow your conviction that not that you've figured out how to do it, but that the science, the technology will lend itself if the prize is big enough, if the problem is big enough. So I think if you look at that, and of course what was interesting was RNA and LNP is what enabled a lot of the CRISPR solutions that are being taken out today. It's enabling now tRNA-based therapeutics, all sorts of other things. And so I think this idea of thinking about a worthwhile big space to unlock and then figure out how to do it regardless of what the approach is, that I think is kind of an interesting flip of the academic model, which is we'll do the work, we'll publish papers, and then these entrepreneurs or big companies are going to come and take it forward. That translation journey is very, very hard. When it succeeds, it should be celebrated by society because it's extremely low probability, because people just give up. Like technology gets in the way, money gets in the way, regulation gets in the way. The question is, can you think of where you want to end up and come backwards and say, okay, what of all the things that we think can work, can we extend and make real? This kind of reverse translation. That's how RNA came into existence, at least in our hands. And now that we've put in that effort, we hope that there'll be many fruits. But I should say one last thing. I mean, another thing that the Moderna experience showed was that you could, in fact, go with biotechnology, which is usually viewed as this slow, low likelihood, really expensive technology. You could go in 6 months to having a vaccine for 1 billion people. If the need is there, but more importantly, if the collaborations are there and the whole system wills it into existence, the capability is there. So the question becomes, why does it now have to take us 7 years to develop the next version of a cancer drug? Why does it have to take us 10 years to develop the next gene therapy? So, you know, it's too bad that this all got politicized, but there is hope, because just under our nose is another biotechnology revolution, that has reached millions of people on the way to reaching hundreds of millions, which is the GLP-1 treatments. That is a strict output of biotechnology. We understand mechanisms we didn't know about, and in a relatively short time frame, 3 years since approval, you're looking at $10 billion markets going to a $100 billion market. That will have, it's expected that about 9% of US population by 2030 may well be benefiting from GLP-1 drugs. I'm not involved in that, so it's somebody else's technology. But I am heartened that one example, people said, oh, no, but that was just COVID. 2 examples in now 2 companies. How many examples do we need to say, you know what, why does this have to take so long and have to be viewed as so daunting? Why can't we actually expect this process to get to better endpoints if we start with a lot more information, much better models that Daphne will talk about, I'm sure, but an expectation by society that this is not just a gamble and a lottery game, but much more something that we can put engineering thought into and fulfill patient expectations. That's what I see coming out of the last few years of experience.
I'd say definitely there's also the regulatory piece to it, right, Noubar? Because, you know, for 15 years or more, mRNA was in the lab, was in research, and because of the pandemic, we had an opportunity to translate to SARS-CoV-2 and find a solution and then get approval. And we're seeing that there are an opportunity for faster approvals elsewhere. But I think when you have something like the cancer industrial complex, where there's a lot of money coming in from philanthropy, even from, you know, with Biden and his cancer moonshot, you see massive amounts of funding going to this particular indication. The scientists are learning and figuring things out that could potentially be translated across to other indications. But it's just unfortunate that all of the money is going to, not all of the money, but a lot of it's going to one indication largely.
I mean, look, my sense on the regulatory side is we don't want to, people had the misperception during COVID that there were shortcuts taken. It's categorically not true.
Not at all, yeah.
At all. But I think all it shows is that what you thought was a 4-hour marathon could be done in 20 minutes by some other form of athlete. And once you see that, then they go, okay, now we got another marathon. It's supposed to take 4 hours, or for me to take a week. But nevertheless, you know, it's just...
So the standards are changing.
Some of it is just expectation. We have, as an industry, accepted the notion that this is what it is. Frankly, our good friends in the pharmaceutical industry benefit from that because that favors incumbents. It's going slow and having it be worth a lot of money to do favors incumbents. Insurgents, which is startups, things that Aloe's doing, things that many others here are doing, benefit from the system asking why. So here's a question that everybody should ask themselves. Why was it worse that people during the pandemic were dying, and that it's okay that people are dying from all sorts of other chronic pandemics, like heart disease, like cancer, like that? Why is it okay for those people to die and that we can take our sweet time and not otherwise? I don't have a good answer for that. I don't know why one life is worth a lot differently. The economy slowed down and almost stopped during the pandemic, but were we making an economic consideration, or were we making a human life? If we want to favor human life, we should take the learnings from the pandemic, and once in a while, and I agree, Aloe, concentrating our bets on picking winners, really bad idea for government funding to do. I think the startup community can come up with solutions, and then we should create environments within which these things can be tested without the concern that if you somehow go fast or increase your expectations, you're going to cut corners. Those two things are no more necessary than when people do it in other industries. I think there's ways to mitigate that.
Or the fact that they do it that way just because it's always been done that way, right? Mm-hmm. And Daphne, I wanted to ask you about your work with AI, because clearly that is a technology that is transforming the way that information is processed, and therefore, right, should help us accomplish what Noubar is saying. It should help us like redesign, rethink that whole system and say, look, it doesn't have to be a marathon. It can be done in 20 minutes. It doesn't have to take decades. It could be done in a few years.
Oh, 100%. And I would also add to that, it doesn't have to fail 90 to 95% of the time, which is what I'd like to come back to. So, I mean, I don't think it's lost on anyone here that the last couple years have been the years of AI, and everyone's suddenly looking this thing, oh my God, AI is suddenly here, except it's really not that sudden. It's just that we've been living on an exponential curve that's been going on for probably decades, and exponential curves are deceptive because when you're in the early part of the curve, it looks really, really flat. And everyone's like, oh, this is never gonna make any progress. And then all of a sudden, you start to hit the inflection point, and then everyone realizes that we are on this exponential curve, and that's what really happened in 2023 with ChatGPT. So that's great, and I think that what we should ask ourselves when we ask how do we take this incredible technology and apply it to the life sciences is first of all to ask what led to this exponential curve. What are the underlying factors? Because those are the ones that we have to replicate. And the biggest factor by far that's driven the success of AI in the world around us is the availability of literally web-scale data that is used to train the models that led to the ChatGPTs and the llamas and such of the world. And data is that is essential commodity, which is what we need to think about of how we get that if we want to take AI and apply it successfully in life sciences, where, to be entirely frank, it hasn't quite hit that inflection point. We're on the early part of that exponential curve. And so when I think about how do we take AI and apply it to the life sciences, first of all, we should all recognize that it will transform everything. It will transform every single phase of the drug discovery and development process and make it more efficient, more effective, and more successful. But then you have to break down into the specifics of what are those pieces and what is it going to take to get us there, because it's not a magic wand. It's not the kind of thing that you just wave some AI fairy dust and everything suddenly becomes better. You need to actually think about getting the right ingredients in place in order to drive that change. So by and large, you could divide drug discovery and development funnel into 3 phases. This is a taxonomy that most people use. There's the early biology discovery of coming up with the right therapeutic hypotheses. What gene do we want to modulate in which patient population and to what end? That is the beginning of the process. Then you take that hypothesis, you turn it into a molecule in whatever modality seems to be appropriate for that intervention, and it's different modalities for different interventions. We've had lots of discussions about small molecules and large molecules and mRNA and siRNA, and we're going to have, and CRISPR, and we're going to have more of those coming up over time. And then at the end of it, there's the part that is currently the most costly, which is, okay, you push that molecule into clinical development, and how do you get that clinical trial to be successful? So those are the 3 phases. As of now, AI has had the biggest impact on that middle piece, the part of taking a target and turning it into a drug. And the reason for that is because that is the part that has the most data. AlphaFold had the fortune of being trained on massive amounts of sequence to structure pairs that have been measured by multiple labs around the world in multiple species. And it is that incredible data set that enabled the incredibly smart people at DeepMind to create that AlphaFold model and a subsequent progeny. Those data sets are only now starting to come into being for the early stage and the late stage of the process, which is figuring out how to intervene in human biology. I'm going to quote Eric Schmidt, who basically said that AI will be to biology what calculus was to physics, a mathematical foundation that allows us to create principled, engineered predictions of what will happen when we do certain things in the system. We've never had that for biology because it's just too complicated. AI will give us that foundation, will allow us to do what I think is the most transformative thing in the space, which is coming up with a therapeutic hypothesis that will actually be successful in intervening in human biology. And not only will make our clinical trials more successful, avoiding that 90 to 95% failure rate, which is an industry elephant in the room that none of us really like to talk about, but it will also accelerate everything. And I think it is that where the biggest impact of AI is going to come in, is in addressing the selection of the right therapeutic hypotheses, which will also allow us to disentangle the complexity of biology. Realize that Alzheimer's is not one disease. It's multiple diseases, each of which probably benefits from a separate intervention, much as we've done in cancer. And that is what's going to give us the high effect size, high efficacy interventions, and address the many patients who are still living with significant grievous illness and unmet need.
Can I double click on
Absolutely.
this real quick? Because when it comes to the data sets, it's really important that we are all working sort of in silos, and we're not necessarily sharing our information as companies and
not.
researchers. And so in order for there to be a data set that AI can interpret or at least work on to offer better, better therapeutic hypotheses, we're going to have to figure out that piece of it. Maybe it's a blind sort of consortium of data that's coming from different companies and different research institutions and labs, but that seems a little bit far-fetched. I don't know how many people are going to be willing to share in that way. And I guess another thing is, AlphaFold is really promising, but proteins aren't the only things in our bodies, and we're going to need to be getting data sets on lipids, on glycans, on all kinds of other biomarkers that are important. So, it really is just the beginning, but it's exciting. There's so much more to do.
No, it is absolutely just the beginning. I gave AlphaFold as an example, not because it's the end of a journey, but because it's a proof point of what you could do when you have enough data in a particular problem to actually train an AI model. It wasn't like, okay, this is the end of the story. I think that when you look at data collection in other domains, the data that's been collected in entire world history up until now is going to be a fraction of what we're going to be able to collect in the next 3 years. And so it's not so much the going backwards and trying to piece together little dribs and drabs of frankly often low-quality data that have been collected, but really thinking about how you create a data collection effort about human health that will truly unlock our understanding of human health and disease. There have been such efforts, mostly funded by governments, like the UK Biobank is one of my favorites, all of us here in the United States. Those are ones that are going to really help us unlock the complexity, the heterogeneity of human disease, and help us identify those novel therapeutic hypotheses. Without concerted effort and collaboration and funding, we're not going to get to those data sets that are going to be transformative.
Absolutely.
Yeah. Daphne and Sam, I want to ask you about the regulatory part of that. But before that, I want to remind everyone to scan the QR code that you're going to see on the screen if you have any questions for any of our panelists. And I am getting an error message on my pad here, so I'm not seeing them. But hopefully, if they haven't come through, you can resend them. So, Sam, I'm going to come back to you, Daphne, but I wanted to ask you about the regulatory part of all of this. With innovative technologies like CRISPR and with approaches like AI as part of the process, are regulatory agencies like the FDA ready to understand what that means and interpret it and really apply them in the correct way to make sure that the valid technologies are getting to patients and the ones that need a little bit more work are left behind?
Yeah, it's a really important question. Goes back to what Noubar was saying. Why do companies fail in biotech? It's either a money problem, not enough money, which hopefully is getting solved as Pablo raises more money. But there's also a science problem, which is becoming a more deterministic bets that Daphne was talking about, right? We know what we're trying to modify or alter, and I'll give you a couple examples from CRISPR. And third is, this all needs to go through a regulatory body. But what's encouraging is with our approval for sickle cell disease, the patient stories really hit home. When one of the patients who de-identified and went on NPR, a woman named Victoria Gray, and she'd been living with this disease, sickle cell disease, all her life, and you had constant hospitalizations, you had chronic pain, you wake up in the morning, you're thinking about pain. A lot of these patients didn't have jobs because they didn't want to think about a job. They were just trying to manage the day-to-day and didn't know when they were going to end up in the hospital and not be treated. In fact, one of our employees who had sickle cell disease, a qualified PhD who ended up at a well-known hospital and wanted opioids for his pain, couldn't get opioids because thought he was a druggie. And so when you have those disease stories, the FDA and the regulators will say, we'll create special pathways. But that's for those rare diseases and where those patient diseases are. But if you think about the broader landscape, we did the exercise, Noubar, that you were talking about. What is the world we want to live in? What do we want to change, right? We all want to live longer. Not everybody, maybe, but most people want to live longer. And we're developing a medicine that's a one-time injection that will reduce your cholesterol by 50% for life. We don't know what the outcome is going to be, but most likely less heart disease. That's the biggest killer. Cancer is a really big killer. 60 years into the war of cancer, we now have new tools to kill cancer. And of course, what I've seen from the regulatory bodies is, there may be for some diseases like rare diseases and sickle cell that create an accelerated pathway, but for broader based diseases, there's still a bit of show me the outcomes and show me the proof. And the problem with that is those just take a long time. The class of medicines we're talking about now with the Ozempic and everything else with GLP-1s, they've been in development for a very long time. Now, the approval happened 3 years ago and exploded like the exponential curve we're talking about, but they've been in development for over a dozen years. And so why are we denying ourselves as humanity with all these improvements where there's a convergence of technologies, we understand the genetics, we can change our lives fundamentally, and have examples where there's a failing pancreas and there's insulin requirements, we can take an off-the-shelf pancreas that we made with ex vivo cells and give it to that person, right? Why are we denying ourselves that? Because there's a long regulatory path. And some of it's dogma, like we spoke about before. Some of it's actually truly that our systems haven't evolved, and our systems do need to evolve. Sure, it's fine to announce a moonshot for cancer and say we're going to support it with some small funding, but it takes more than that. It takes a fundamental systemic change where the regulatory bodies are coordinated with the research institutions and the companies that are developing medicines to bring about medicines faster in a more effective way to the people and patients that deserve it.
And Daphne, with a technology like AI, where we've seen the headlines, generative AI, and the conflicts and the concerns that it raises, have you had any interactions yet with regulatory bodies in terms of what they would be looking for or how receptive they are to a product that might come from a process in which AI was heavily involved?
So I think that's a great question, and it's important to sort of think about the different pieces of this. When the FDA gets to review a molecule, they don't really care where the molecule comes from, because ultimately the molecule is what's being tested in a rigorous randomized clinical trial, whether it's a scientist dreamed it up at night, whether it came from a screen or whether it came from a generative AI process. Ultimately, it is a molecule, and that is what they review. I think what becomes a little bit more nuanced is when you're asking to review an AI biomarker for defining which are the patients to whom that drug should be prescribed. And I think that is, and I think Sam really alluded to that really well, we have a kind of dichotomy in the industry between rare disease drugs, where it's a small patient population, where there's a lot more, I would say, leniency in the part of and openness on the part of the regulator, versus the large common indications that are held to a much higher bar for regulatory approval. But I think what is really the case is a lot of these large indications aren't really a single disease. They are multiple diseases that are each quite biologically distinct. When you think back not that long ago, 15 years ago, breast cancer was a disease. And now we understand that that is absolutely not the case. And there's a completely different biology for BRCA breast cancer versus, exactly. And so now they each have their own therapeutic intervention for a smaller, better defined patient population. And that is what has allowed us to move away from the lowest common denominator, which is chemotherapy. Let's just kill the cancer cells. And so I think that has been transformative in cancer. And we are heading towards a similar transformation in other therapeutic areas, where we still often think of these large indications as a single thing when they are not. And in order to correctly identify those patient populations, often the biomarker will have some elements of AI in it. And then how do you get the regulator to accept that as part of the triage journey of which patient gets the drug and which one doesn't, so that we can truly live our vision of precision medicine, which everyone's been talking about for quite a while, but we haven't really seen it come into play anywhere outside of oncology. And I think that is where the future is, and it is that the regulators are going to have to come to terms with.
So I thought I would share with the audience a couple of I talked about Royalty Pharma and how we fund the ecosystem at Royalty Pharma. We've been doing that for 27 years. But totally different perspective, another hat that I wear. Share with you a couple of interesting situations that I'm involved with, which really highlight how things can be accelerated and also convergence. And it's actually, talking about acceleration. So at NYU Langone, a group of transplant doctors developed a model, which is a model of a decedent. This is a brain-dead human that had donated the organs for research. And what they decided, and obviously with the consent of the family, that the whole body can be used for research. So we are about to embark on a collaboration with NYU on another company that I'm involved with personally, where David Sachs' 40-year project, one of his 2 life projects, developed a colony of pygmy pigs. And over time, inbred these pigs to knock out genes that cause rejection in humans. So we now have a small pig that is able to produce organs that are the right size for a human. Everyone else is working with bigger pigs, where the organs get to be too big. And so we have an edited pig where the genes were knocked out to cause rejection and with the right organ. And we're about to start, maybe in this month, to put those pygmy pig kidneys into a decedent to see if we can actually improve, actually in that case it's for transplantation to actually, eventually figure out if we can actually have these organs for real humans. And the plan is to maybe, do 20 or so of these transplants over the next 18 months. And if we succeed, and the initial goal is to see if we can sustain, the decedent with a pig kidney for 2 months and monitor what's going on because these pigs have been edited. It's going to be probably the kind of immunosuppression that is used today when you go from human to human, and then to start to extend that. So that's one interesting thing. And what's shocking to me is how this model of a decedent, which is a person that is no longer alive, brain dead, allows for experimentation in a real human with a pig organ. The second example is another technology that I've been involved with for the last 4 years. Actually, a partner of mine who's in the audience, Eric Berglund, and I were having a conversation. He's a transplant surgeon, about what is something that would really transform transplantation. And he said to me, a normothermic perfusion pump. So this is, instead of an ice box where you can put an organ, this is a pump that can actually maintain an organ with circulation at body temperature. And 4 years of investment with Dean Kamen developed this device. It's now fully developed. We got breakthrough designation from FDA last year. And now this becomes a platform. So talking with Sam earlier this year, we said, okay, what if we take your technology, CRISPR, and we take an organ, we're starting now with pig organs and potentially discarded human organs, and then see if we can modify these organs with CRISPR to actually cloak the organ so that when you put it into a human, the immune system would not see the organ. It becomes invisible to the immune system and potentially not require immunosuppressants. But what was interesting in the conversation we had at the beginning of this year was his realization, where he said, wow, with CRISPR, we always think about doing something, but you end up impacting the whole body, right? And with potentially consequences. What if we just treat an organ, right, that has a problem by using our perfusion pump, and we're just locally, treating the organ and not the whole body? And I think FDA is going to be a lot more open to that than essentially applying CRISPR on the whole body. And the last thing we're doing with organs is with another approach, which is the Yamanaka factors, which, a Nobel Prize scientist took a skin cell and took it back to a stem cell. And it's been shown that if you use Yamanaka factors, you can actually maybe rejuvenate organs. So again, what we're doing there, we have the perfusion pump. We're going to take organs from a decedent, take one kidney out, put it in our pump, apply the Yamanaka factors, and see if we can rejuvenate the kidney of this human. And it's a kidney that's working, right? The person is brain dead, and then put it back into the person and see if we're able to improve kidney function. So convergence, you're using a device to actually, treat an organ that's failing, and then But using, again, this decedent model, which is brilliant. It's amazing that this team at NYU was able to develop this model that allows for experimentation in a body that's working. It's a brain that, individual, but the body's working, and you can do all sorts of really cool things. Anyway, that's exciting stuff that...
So I want to raise we have a couple of questions from the audience on issues of scalability and cost, which are related, obviously. Sam, let's start with you. How do we make a treatment that is CRISPR-based, which right now is quite expensive, more scalable and more available to more patients?
It's top of mind for us. I grew up in India, and there's actually tenfold more patients who suffer from sickle cell disease than in the US. People didn't realize this, but if you go to China, the amount of people suffering from thalassemia is huge. And Africa, obviously, is where there's the most patients who suffer from sickle cell disease. And so how do you get these medicines to patients when in the US, the current price of the drug is about $2 million? And the beauty of what we're doing in biotech is that they start off expensive, but the social contract here is that as things get genericized or as technology improves, they all become lower cost in the end. Now, we talk about in vivo editing, for instance. Can you directly edit these cells inside the bone marrow with the virus or with the delivery solution? That can actually go to patients with sickle cell disease at a $10,000 price point or $20,000 price point. We have now done this in cancer. The autologous CAR Ts, which have cured many patients now with heme malignancies, they cost about $500,000 in the US. But we've now manufactured allogeneic, which is that instead of taking mini pigs, we're taking healthy human donors. We take their cells and we modify these T cells to make CAR Ts. And that cost of manufacturing has come down 2 orders of magnitude into the thousands of dollars range, like an antibody. So I think technology improvement is one of the biggest levers in reducing costs. The other, though, obviously, is as more competitors come in, there's newer companies that figure out how to do this on a lower cost. Whether it's in the US, whether it's in Asia or other places, you are going to see this pressure in cost, and eventually things get genericized and they become lower cost. But the problem is, at first, as a society, we do have to pay for it. If you don't, there's no incentive to put money into a long bet. I mean, as Noubar was saying with Moderna, it was what? Billions of dollars to take on a bet that this would eventually be useful for society. Who is going to take that long bet if there's no returns in the early cases? And so that model is something that I think should be espoused by society in this era where everybody's talking about drug pricing, where initially, as technologies break through, you have the payment models, you have the ability to pay back the investors that put the money into it. But eventually, operators like us have to have a firm view that the costs need to go down, not just a little bit, but by orders of magnitude to make it available globally.
So costs need to come down, but I think there's also a change in perspective that's needed, right? And because currently insurance companies will look at $1 million plus therapy and say, why should we pay for that? Because they're not really looking at kind of the ROI on that, the longer term perspective, which is not how we've historically reimbursed for disease treatments, right? It's been a little more kind of pay as you go. Do you see the needle moving at all as we get into this era of cell-based treatments that are going to be a little more expensive up front, but perhaps over the long term, if we look at, Pablo, kidney patients, if we look at people with sickle cell and the healthcare costs that they incur over a lifetime, is it cheaper and does it make more sense to have a higher upfront cost? But that's the kind of calculus I think that we're not quite there yet for
well, I'll let Pablo answer, but one thing I'll just say, fact with sickle cell, we got so much scrutiny, and everybody did this ROI calculation to say, what is the pharmacoeconomic cost? It turns out our system's paying about $300,000 per patient every year because they have so many hospitalizations, they have so much pain, so many effects. So it was like a no-brainer at the end of the day when they did the calculation. But at the end of all that, it's not just pharmacoeconomic. This was not even considering the value to that person's life. And what one of the Medicaid agencies told me was very striking, which is they actually want to get these patients off of Medicaid and into society working and be productive, right? So if you can get these patients cured and not think about their disease and think get a job, they actually get off Medicaid. So to me, this is not a question. I think there's a lot of articles written about it, but the ROI is, is there, the pharmacoeconomics are there, but most importantly, the value to patient's life or person's life that you're completely changing, it's hard to calculate, but it's invaluable.
Yeah, I think the innovation is also occurring not only in the science itself, but in manufacturing, and it's driven by competition. And we have seen significant declines in cost. And for example, CAR-T, where it was $500,000 or more, and it's coming down. And I've heard recently people talk about sub 100,000 treatments. So we'll see. Another interesting data point, for example, thinking about this other thing I'm involved with pig organs, is that they're very inexpensive. Every time, I mean, you get 6 or 8 pigs from every litter, right? So potentially, manufacturing these organs is going to be not expensive at all. And, will that work? We'll see. But we think we might be in humans, real humans, in a trial in 18 to 24 months. But innovation in manufacturing is really key. And it is actually, it's so interesting to see how, different technologies, different modalities, different approaches, all of a sudden, bring down costs in a very, orders of magnitude.
If I could just add very briefly, I think it's also important, and Sam alluded to that, to look at the other side of the equation, which is the value provided to the patients. We currently pay often hundreds of thousands of dollars for a cancer drug that extends your life by 2 months. It doesn't fundamentally alter the course of the disease or the final outcome. It gives you, not to diminish that, but a couple of extra months of life. So what about drugs that truly are curative, truly modify the course of the disease and dramatically change your outcome? Those are worth a lot, and I think it's important that we continue to highlight that.
That's what I'm excited about. It's canvassing the landscape of scientists that are working on new modalities. And at Major, the concept of peptoids, this class of peptidomimetics, where you're basically improving the peptide. It's the right side. It's like a Goldilocks drug lead, because you've got serum stability, you've got non-immunogenicity, you've got highly target specific, so you're reducing and minimizing the potential for off-target effects, room temperature stable. Just finding ways to make it more economical. You're also finding ways to make it more efficient right at the beginning of the cycle of drug discovery and development, which takes you a long way, especially when you get into the clinic and you're looking at potentially failing after you've done your risk assessments in phase 1, potentially failing because of off-target effects. You've got to try to solve for that at the beginning of the life cycle of the drug development. I think that's why I'm excited to be involved in this particular class of molecule, peptoids.
Now, Noubar, we have a question here about the coordinated effort that companies like Moderna and Biotech have with the UK government, which of course has a completely different healthcare system. And without a national health system like the UK has, how do we make sure that the coordinated government effort that we saw, as you mentioned, with the COVID vaccine, with Operation Warp Speed, how do we ensure that that continues, or the spirit of that continues?
Well, I think a lot of the current coordinated efforts have to do with the cancer vaccines, because I think that the big beneficiary of the technology expansion and scale will be actually the use of these same approaches in cancer vaccines, which are proving quite encouraging, let's say, and we hope by the end of this year, early next year, there'll be definitive phase 3 data that begins to start emerging. And that could be a total game changer, but of course it's piggybacking off of the fact that we now know there's a safe way to put in antigens. We're not putting in spike protein antigens, we're putting in cancer. And in melanoma, we've seen 40–50% improvements over Keytruda, and we're beginning to get into other ones as well. But to your question about what role can government play, again, it may sound naive, but if we decided to go after some disease area in an intense collective way. So what happened during Operation Warp Speed? First of all, the clinical trials that were needed to be run in parallel were facilitated and put in place with networks that were established overnight. So it turns out that the HIV clinical trial networks that existed in the US for a long time were very quickly repurposed to do this in an intense way. The FDA went from sitting back and asking questions to actually coming up with the questions that needed to be put into the clinical trial design, which in peace times, when it comes to drug development, is not allowed, but I still can't figure out for my dying mother why that's a better answer. And so again, I think most people who make regulations don't actually realize that when if it applied to them, they'd want it to be done differently. So there was that coordination. The capital, what did the government do capital-wise? They basically said, look, we need a solution in an unusual time frame. We didn't know if we could develop a vaccine in 6 months or a year or 2 years or 3 years. We knew one thing, every day the economy was losing gigantic kind of value. So basically they said, look, we'll do two things. One, we'll facilitate the actual cost of scaling up because no one company was going to do that on its own nickel. And two, we will send the signal from the market. If you can do this, this is how many doses we'll buy for this price. Now, if you did that in other significant areas, as uneconomical, uncapitalistic as it sounds, not to solve the problem in a nationalistic way, but actually just prime the pump, I think you'll see certain things advance much faster. I think that risk capital will be attracted to more certainty of the prize versus what we deal with now. Uncertain science, uncertain technology, uncertain products, uncertain clinical trials, uncertain reimbursement environment. It's a wonder we ever work on this. Uncertain market size. It's a wonder anybody works on this. We saw an instance where that can be completely altered when some of the uncertainty is mitigated, because what they did is they took the capital risk and shared it with the innovators. It's too bad that COVID got politicized, because we took everything that could have been learned from that and hid it, lest it somehow be used for elections. And yet, I think it was the best moment of biotechnology. And in an interesting way, it's being followed by another example. I think we can do more. I really wish politicians would realize that they're going to get a lot more votes when they move the needle. And by the way, to me, the only way of dealing with healthcare costs, and Daphne mentioned earlier, is not only not to only focus on the last 2 months of life, but to move way upstream. So we work quite a bit these days on what we call preemptive medicine. That is, do we have the wherewithal to detect disease before it manifests altogether, to intervene? If you say that to regulators today, they'll say, what's the disease? And you go, well, if you have the disease, it's too late now. We'd like to go upstream of the disease. And they'll go, well, what's the disease? And so prevention, which we do with vaccines, could also be done with molecular interventions, not vaccines, that actually delay the onset of disease. How do you get that approved? It's tricky. So I think we're going to have to do all this with better information, better models of living humans, digital twins, all the things that are being bandied around. I hope the end result is that we dare to put, on the one hand, avoidable deaths, and on the other hand, the concern that if we did this somehow in a new way, we might be taking on new risks. And I think society can make that call.
And I think with the new technologies, it feels like we're coming to that tipping point where we're forcing that.
Well, by the way, countries like the UK, because that was the origin of the question, countries like the UK, frankly, because of their massive focus on the cost of healthcare, have to, in a way, lead in the way. You can't both say, I'm not going to use, quote, expensive drugs, and I'm going to make your development even harder than it is in other places. So I think they've been much more data-centric. Their regulators are beginning to say, let's think about talking ahead of time and not thinking we're adversaries. So I'm hopeful that the UK and other places will play a leading role there.
Actually, to just jump on that, the UK has been also world leading in the data creation ecosystem for understanding which patients are going to get disease and what are some of the underlying causes of disease. So they've been a huge driver for innovation in the healthcare space that I think has been enabling for many of us. So I think many governments could stand to learn from the UK in terms of how do you think proactively and not reactively about healthcare.
And Daphne, I wanted to have another question for you as well, which is about the data that's collected. And you mentioned, in terms of the data that we currently have and that's existing, a lot of it is based on European populations, white European populations. How can you account and include a more diverse population without going out and gathering new data? Are there ways to model that? How is your company dealing with that?
So I think that's a really important question and one that we think about a lot. I think that currently the number of disease-modifying treatments is so limited that I don't think we can extrapolate from to what extent is a drug that is effective in a European population, is that also going to be effective in other populations? So far, the evidence, such as it is, actually seems to be supportive that the fundamental human biology is shared, even if the prevalence of different diseases, sickle cell, of course, is a great example. It manifests in different proportions in different populations, but the underlying biology is the same biology. So if you find a treatment, the treatment works in patients pretty much across the board. And so I think what's really important is to disentangle the underlying biological mechanisms. And I think once you do that, you will find effective therapeutics. Having said that, I think having more diverse populations not only increases our ability to make sure that our treatments work in a broader patient segment, but also actually powers discovery. And what we've seen is, for example, that there's much greater genetic diversity, for example, in people of African origin than in European because of the population bottleneck that existed there. So you can make remarkable discoveries by looking at the genetics of people from diverse ethnic lineages. And so I would really encourage all of us to think about how we can collect more of that genetically diverse data so that to empower the next set of discoveries.
Well, thank you, everyone. I want to thank all our panelists for giving us an insight into where we are right now with biomedicine. It's a very exciting time. Thank you.
Thank you.
It's in a